So this morning we've asked Christof Koch, the Chief Scientist and President of the Allen Institute for Brain Science, to kick off the day with some introductory remarks about the Institute for Brain Science.
Thank you very much. So yesterday and today you've seen results of the voyages of our Starship, the Allen Institute for Brain Science, our 10-year mission to explore new worlds, to seek out new life, and to boldly go where no one has gone before. And so we are nearing the end of the 6 years of this 10-year mission. And so it's time to check in on sort of at the high level some of the insights we've had. We started off many years ago with this metaphor, this analogy, that we're trying to understand a very complex system, and there are different ways how you can try to understand complex systems. So on the right, we have a tool that's more or less part of our body now. It's almost like, well, it's basically an extension of our body, and we seek to understand it, and we can understand it at different levels. We can understand it at the level of the app, of the application. That's the one most of us care about. We can understand it at the level of the algorithms that a few people care about. Any one of those apps has a series of algorithms and computation. Or we can seek to understand it at the level of its individual components. So here we know there are particular transistors, all based on transistors, and there are a few types of transistors. They're like power transistors, they're switching transistors, the N and P doped, etc. There's a handful of individual components. On the right, we have an organ of the body, like the liver, like the kidney. It performs its physical function. Its main mission is to make sure that the DNA of its carrier is passed on to the next generation. And it's also the most complex piece of highly organized physical matter in the known universe. And so just like you can look at the cell phone and the apps and the algorithms and the component, this is also a popular way to analyze the brain. This was particularly popularized by Herbert Simon and then later on by David Marr in his multiple levels of analysis of any computational system. So you can analyze it at the high level, at the behavior level, at the app level, at the cognition level. If you look at things like visual perception or memory, or you can analyze it at the level of the individual components, or you can analyze it at the level of computation. So today I just wanted to sort of highlight, given what particularly the last talk yesterday, the masterful talk of Ed, wanted to highlight what can we say about the components. Who knows this statue? It's actually here right at the SAM. It's due to a German sculptor. She's called Katharina Fritsch. It's called Mouse and Man. Sort of more like a nightmarish vision. So here I have the brain. So it's a life-size replica of a human brain, and here I have an over life-sized replica of the mouse brain, just to remind us that those are the two objects that we seek to understand. Now, it's only in relatively recent time, it's only less than 4 centuries ago, with the discovery of the microscope, a tool by Anton Leeuwenhoek in Amsterdam, that people discovered, Robert Hooke discovered in London a couple of years later, by looking at a piece of cork, there are these little units called cells. And then he went on to look at living plants and microorganisms in water, and he published sort of the Allen Cell Type Institute database of its time, namely Micrographia, a very influential book. And then 200 years later, a triptych of German biologists, physiologists, Schleiden, Schwern, and Virchow sort of established what today we know as the cell, the 3 tenets of cell theory. In other words, all biological organisms consist of 1 or more cells. Cells are the basic unit of biology, and cells beget other cells. And then 50 years later, so this is the beginning of the last century, 1909, our patron saint, as it were, Raymond y Cajal, published a book Histologie du système nerveux de l'homme et des vertébrés, where he explicitly called out the nervous system of human and other vertebrates, in which for the first time sort of he described the great variety of different types of neurons that he found using the Golgi stain in the brain of human and other animals. And so we are beginning to realize that they aren't just sort of brain cells, although it took computational people another 100 years to realize that brain cells aren't just a homogeneous soup, but they're actually different types of them. And so ever since then, since roughly 110 years, we're trying to understand how many different types of neurons there are actually. So this is a cell type problem. This is from a beautiful article, a really magisterial article written by Hong Kui and Jeff Sainz, in which they describe sort of how to go about classifying cells and looking for cell types. Of course, this revolves around the point, well, how do we define cell types? Are there cell types? How real are cell types? Of course, it's part of the human visual system, the way we process information, that we cannot but help finding commonalities in whatever we look at. So we believe that we can treat the brain not just one neuron at a time, which would be a hopeless task given the 86 billion neurons in the human brain, but that they're actually classes, and so we can sort of do a dimensional reduction, as it were. Rather than trying to understand a brain of 86 billion neurons as a human brain, maybe we can understand a brain that consists out of a certain number of cell types. And there are various ways we can classify them using morphology. That's a traditional one, Raymond y Cajal, doing sticking microelectrode into a neuron in a slice, albeit sort of under these artificial coma-like condition, is a different way to doing it, as we're doing it in IVSCC. And the way we've also been pursuing, of course, big time over the last 10 years, is by looking at the molecular signatures. Ultimately, we like to link it to function, i.e. to specific behaviors. That is going to be an extremely challenging task, and it's probably going to take us the next century to really systematically link the different cell types to ultimately their function in behavior. It's going to be very, very difficult because the system is highly robust and highly redundant, so knocking out any one type may not lead necessarily to specific loss of function. And of course, cell types can be involved in many different computation. Remember, it's not of the business of the brain to be easily understood. The business of the brain is to pass on the DNA of its carrier. So this is a point I like to stress. I like to stress the similarities. Ed, and he showed the slide also yesterday, like to stress the differences. You remember at this all staff half a year ago, where I showed you, by removing the scale bar, I showed you human neurons and mouse neurons, and people basically were at chance. That's not to say that we couldn't be trained to distinguish human from mouse cells, but at least under the condition when I just show you one after the other, the stuff is basically very, very similar. Now ours, of course, is bigger because our brain is 2.5 millimeter thick, our cortex, while the mouse brain is only 700 micrometer. And so the question is, Ed, for example, mentioned yesterday that human cells are more complex. The question is, are they just more complex because they're 2.5 times thicker? So in other words, they have 2.5 times more spines, for instance. Or are they intrinsically, for example, is the Hausdorff dimension, the embedding dimension of the dendrites, is that higher in human and in mouse? At this point, we don't really know. But sort of, they're remarkably similar. Here we have two beautiful cell pair. On the left, you have a layer 5 mouse, and on the right, you have a layer 2-3 human Martinotti cell. That's the scale. So really very beautiful cells that are overall remarkably similar in mouse and human. Of course, there are lots and lots of little differences. We know from electrophysiology, this is from Jonathan Ting and his team. If we look at so he is plotted the electrophysiology of human cells from human middle temporal gyrus surgical material intermixed with mouse V1, and there are all sorts of differences, minor differences, I would submit. Overall, spikes is a spike. Whether it's a human or neuron, they adapt. They have all of the basic currents that we have in mouse, with lots and lots of little differences. And this is a summary of sort of a very large fraction of the Institute's resources, and people are engaged in this at the intellectual level, at the physical level, at the logistic level, analyzing the transcriptional properties of individual cells. On the right, it's from mouse visual cortex. You saw it yesterday on some of the posters. On the left is from human temporal cortex, as Ed presented. What's remarkable about it is, what I stress, what I find truly remarkable, is the overall great similarity between these taxonomical trees. 10% of all the cell types, whether it's a mouse V1 or whether it's human MTG, 10% are non-neuronal types. We know roughly there are half of all the cells in any one brain area in this cortex are non-neurons, half are neurons. In terms of cell types, they're 10%. And then if you look at, specifically the breakdown of excitatory and inhibitory at the class level, they're very, very similar, right? You have the NDNF, you have VIP, P-valbobine, you've got somatostatin, both in the mouse and in the human. There are lots of subtypes, but overall, it's roughly the same number, sort of about 80 or so cell types in the mouse and in one area, and about 80 cell types in another cortical area in the human. Now, mind you, these aren't matched, right? So it's V1 in the mouse and middle temporal gyrus in the human. Ultimately, we'd like to match them. Remember, the last common ancestor between those two brains, the last common ancestor goes back 80 million years. So that's a lot of iteration of the evolutionary clock that distinguish this and that brain. Now, what's really interesting that overall there's a great deal of similarity between mouse and human, but the details differ. Right? We've seen that also in the viruses that we're trying to build that sort of are selective for mouse, and then trying to transfer those viruses to a human tissue, that at the detail leaves level, so at the level of the bottom of this tree, the specific genes that are characteristic for the individual cell types may differ greatly. And of course, this is really critical if you want to build drugs, or if you want to have an understanding of the details of the way humans process information in the brain, in particular in health and in disease. And of course, this observation that the details differ greatly helps to explain why billions of dollars and 20 years of research by big pharma into mouse or rodent pharmacology and receptor studies has not yielded any great new drugs, because the details differ between us and mice. The difference between that and that brain, in terms of physical difference, is roughly a factor 1,000 in terms of mass. In terms of numbers of neurons, pretty much exactly a factor 1,000. Even if you go into cortex itself, it's a factor 1,000. So then why is it that we're never going to have a conversation, we're never going to have a mouse attending this conference, at least not as an active observer, as an active participant, I should say, maybe as a passive observer? Why is it, given this great deal of similarity at the level of the hardware? And here I think it's really interesting to look at the neural network revolution over the last 20 years. So this is, for example, Google's Inception network that you may have seen in their dreamscapes, in these fantastic imagery, when they take a picture or a series of pictures, a video, and then transform it in this hallucinatory manner. This uses this network. That what we've learned over the last 20 years from deep convolutional networks, that ultimately, of course, are intellectually descendant of neural networks, that was first described by Hubel and Wiesel and by Fukushima in the visual system of cats and monkeys, that really there's a big difference between, if you have a similar level of representations in terms of simple neural networks, the more layers of, the deeper your network, right? The deep convolutional network refers to the fact that there may be 10, 20, 30, modern networks may have 100 or more layers, that the deeper these networks are, the higher level of abstraction, right? So simple networks can do simple abstraction, like, between lines of different orientation, and as you add more and more features, you can make more and more high-level distinction, including among 1,000 different breeds of dogs, something that most humans are incapable of. Or you can even do more abstract computation, such as if you want to play a game of chess or Go or shogi, where you can where these networks can learn to represent very, very abstract patterns. And so the lesson here is, the deeper the network, the more powerful the computation the system as an overall performs, although the individual components are all very, very similar. And so the hypothesis that I think, or the proposal that's beginning to emerge, at least for me, from all this research that we've been doing here over the past 6 years, is that it's about cell types and the difference between cell types and humans. So many of you probably won't know, because it happened before many of you were born, but in the 80s and 90s, it was confidently predicted that the number of genes in humans, of course, as a matter of fact, is going to be much higher than in the mouse. In the mouse, people estimated 20,000. And for humans, I just went back and looked where people predicted 50,000, 80,000, 100,000 or more genes. Well, it turned out, as we all now know, it's roughly on the order of 20,000, whether you're a mouse or whether you're human. And I think likewise, if I look at this data generated by our teams, it really looks like that overall the number of neuronal cell types will be roughly the same between humans and mouse, roughly. Right? That's not to say that there aren't going to be some unique types, but it's going to be it's going to be very similar, and of course with evolutionary specialization, because they did evolve in these separate ecological niches over the last 80 million years. And the key difference that I always like to emphasize, that people don't like, because people are always looking for magic bullet to explain why we're so different, is the fact that we have 1,000 times more processing resources than the mouse, and this enables us to come up with completely new invariances and abstractions, such as we have in language and in very abstract ideas. And here, I just wanted to end on this note. There's this really interesting metaphor, particularly in light of the fact of our donor, because let's look at the difference between biological and technological evolution. So here we have the two brains. And on the right, I'd like to focus on one processor. Who knows what a 6502 is, or its close cousin, 6507? Okay, very few people. All right. So this, at its heyday in the 40 years ago, was the first MOS system, the first commercially available, relatively cheap microprocessor, 6502. And in fact, our benefactor, Paul, programmed famously BASIC together with Bill Gates. They compiled the BASIC from the Altair onto the 6502, the BASIC and its basic commands. I mean, the language BASIC, which is really at the core at the time of Microsoft. So you look at the 65, and it also powered the Atari video game. And so it really powered that entire homebrew computer club mentality, and the first video games, and the first microprocessor that was cheaply and widely available, and then its twin, 6507. Now it had a stunning, it was 3 megahertz clock. It had 3,500 transistors, and it used 8 micrometer with MOS technology, okay? Now we fast forward, 40 years later, we have the A10. This was for the iPhone 7. Now, of course, it's already been superseded by the next one, but the A10, all right, uses the same basic technology, but now the resolution has shrunk from 8 micrometer resolution to 16 nanometer, factor 500. Its clock has sped up from 3 megahertz to 2.5 gigahertz. It's a factor of 1,000. Its parallelization has increased from 8-bit to 64-bit, so factor 8 between those two. And it has multi-cores and has multi-threaded and all the newfangled stuff that's on all computers today. So the raw computational power is at least a factor of 10 billion difference between the 6502 and the A10. Now the 6502 you could talk to, you could scream at if, for example, your video game didn't really work very well, but it wouldn't talk back. Now to your iPhone, of course, you can talk and it'll talk back, right? But the point I'm trying to make is same thing here, right? The difference here is, okay, it's 1,000 neurons. We don't know the raw computational difference in terms of raw computational power between the mouse and the brain, but this generates language. It speaks and you can talk to it. This one doesn't. Same thing on the right. That one doesn't talk. This one does talk. And where's the difference? Well, at the level of the metal, it's still CMOS, it's still transistors, it's still NAND and NOR logic. So all of that, it still has a bus. It has all the same features in both cases. But here, of course, you've got 40 years of technological evolution. But the basic hardware is really very, very similar. And so I think that's going to be the case for the difference between that brain and that brain. And with that, I thank you for your attention.